This study, titled Effect of Refrigeration, Room Temperature, and Processing Time on Serum Immunofluorescent Antibody Titers for Sarcocystis neurona, investigates the stability of immunofluorescent antibody, IFA, titers for S. neurona in horse serum samples under different storage conditions. The research is motivated by the fact that timely and proper refrigeration of serum samples is not always feasible in veterinary practice, especially when samples need to be transported over long distances or when veterinarians in certain regions do not have immediate access to specialized laboratories. The study seeks to determine if serum samples can be reliably tested even if refrigeration is delayed or not possible. This is especially relevant for diagnosing equine protozoal myeloencephalitis, EPM, a neurological disease in horses caused mainly by S. neurona. The diagnosis of EPM often begins with evaluating serum antibody titers, though a definitive diagnosis requires confirmation of protozoal infection in the central nervous system, CNS. The determination of serum antibodies can help estimate the likelihood of EPM, rule out infection, or conduct epidemiological studies. The study focuses specifically on the stability of IFA titers, a common method used to detect antibodies to S. neurona, under various storage conditions. Introduction. Equine protozoal myeloencephalitis is a significant neurological disease found in horses in the Americas. While S. neurona is the primary cause, other pathogens like Neospora huggisi and Toxoplasma gondii have also been implicated. The clinical signs of EPM vary depending on the affected location within the central nervous system, including the brain and spinal cord, and can manifest acutely, subacutely, or chronically. Diagnosing EPM involves a combination of neurological exams and immunological tests. While post-mortem examination provides a definitive diagnosis, ante-mortem diagnosis relies on clinical signs, ruling out other diseases, and detecting intrathecal antibodies against the parasite. Immunological tests like the immunofluorescent antibody test, IFA-T, and S. neurona surface antigen, SNSAG, immunosorbent assays, ELISA, are often used. Because collecting cerebrospinal fluid, CSF, has inherent risks and costs, veterinarians often start their diagnostic process by evaluating serum antibody titers for S. neurona. Serum antibody presence does not confirm clinical EPM, but it is useful for assessing the likelihood of the disease, ruling out infection, or conducting epidemiological studies. In practice, serum samples are usually refrigerated for transport. However, maintaining a cold chain is not always possible. Many samples are sent to the United States for testing from locations lacking specialized diagnostic labs, which can delay analysis for several days. Pre-analytical factors like temperature and storage time are known to affect immunological test results. This study is the first to evaluate the impact of refrigeration and room temperature on serum antibody titers for S. neurona in horses. The main hypothesis of the study was that IFA in serum samples stored at room temperature in temperate weather would be stable for at least 5 days. Materials and methods. This research employed a prospective longitudinal study designed to compare IFA titers from serum samples stored at room temperature, 20 to 23.3 degrees Celsius, or 68 to 74 degrees Fahrenheit, and at 4 degrees Celsius 39.2 degrees Fahrenheit. The study included 3 different time points: Day 1 baseline, the day of collection, Day 5, and Day 10 after collection. Serum samples were obtained from 22 horses previously known to have IFA titers for S. neurona. For each horse, 10 milliliters of whole blood was collected in red top tubes and centrifuged after clotting to separate the serum. 2 1 milliliter aliquots of serum were collected from each horse. One was stored at room temperature, and the other was refrigerated. The room temperature aliquot was tested immediately for S. neurona antibodies using the IFA-T method. Both aliquots were retested on days 5 and 10. The IFA-T began with serum dilutions of 140, and the endpoint was recorded as the last dilution showing clear fluorescence of the whole parasite. The researchers used the endpoint titers to make comparisons, which is important because clinicians calculate serum to CSF IFA ratios for EPM diagnosis. A low IFA-T ratio of less than or equal to 64 is considered an indicator of EPM. The study was approved by the University of California Davis Institutional Animal Care and Use Committee, IACUC. Sample size calculation indicated that a minimum of 14 samples was needed based on a determined effect size, which is smaller than the 22 samples that were included in this study. SAS software version 9.4 was used for statistical analysis. The Shapiro-Wilk test was used to check for normality of the sample. Because the sample was normally distributed, a paired t-test was utilized for comparing antibody titers across the different time points. The IFA t-titer was the dependent variable and the independent variables were time point and storage condition. The aim was to compare IFA t-titer differences over time and under different storage conditions. Results. The study involved serum samples from 22 horses, including 14 geldings and 8 mares, mostly of thoroughbred breed, with an average age of 12.5 years. There was no statistically significant difference in IFA titers from baseline to day 5 and day 10 for samples at room temperature and under refrigeration. Further, there were no differences in IFA titers at each time point between samples stored at room temperature and those under refrigeration. Specifically, comparing the serum IFA titers for S. neurona at baseline with samples stored at room temperature for 5 days showed no significant difference, p equals 0.741, 95% CI, minus 56.83, 78.65. Likewise, there was no significant difference between baseline titers and samples stored at room temperature for 10 days, p equals 0.677, 95% CI, minus 50.01, 75.46. There was also no statistical difference in IFA titers at room temperature between days 5 and 10, p equals 0.949, 95% CI, minus 57.50, 61.14. When considering samples stored under refrigeration, there was no significant difference between baseline titers and samples stored at 4 degrees Celsius for 5 days, p equals 0.964, 95% CI, minus 81.81, 85.45. Similarly, there was no significant difference between baseline titers and samples stored at 4 degrees Celsius for 10 days, p equals 0.573, 95% CI, minus 109.4, 62.15. Additionally, there was no statistical difference in IFA titers at 4 degrees Celsius between days 5 and 10, p equals 0.5, 95% CI, minus 102.6, 51.67. When comparing samples stored at room temperature with samples under refrigeration, no statistically significant difference was found between the two storage conditions on day 5, p equals 0.688, CI, minus 55.51, 37.33, or day 10, p equals 0.104, CI, minus 80.8, 8.07. Discussion. The study's findings indicate that immunofluorescent antibodies for S. neurona in serum are stable for at least 10 days, regardless of whether the serum is stored at 4 degrees Celsius or at room temperature. This is significant because it suggests that results obtained from unrefrigerated samples or samples not tested right away can be reliable. Pre-analytical factors, including the steps before the lab test, have been classified as either technical, due to sampling and handling, or biological, inherent to the animal. While this pre-analytical phase is important, limited research is available for pre-analytical conditions in both human and veterinary medicine, particularly related to serum antibody measurements in horses. Previous studies in horses and humans have reported the stability of immunoglobulins for up to 72 hours at room temperature and 6 days at 4 degrees Celsius. Another study found that IgA antibodies in serum remain stable for 48 hours at room temperature before centrifugation and 10 days at 5 degrees Celsius after centrifugation. The current study had certain limitations, such as the lack of evaluation of IFA for other EPM-related pathogens like N. hugesi and T. gondii, and the lack of CSF antibody analysis. The samples in this study were centrifuged immediately after coagulation, which is not always the practice by field veterinarians. The environmental room temperatures for the study ranged from 20 to 23.3 degrees Celsius, and therefore, the effects of other environmental temperatures on the stability of antibodies were not examined. Despite these limitations, the study's main conclusion is that serum IFA for S. neurona is stable for up to 10 days at both room temperature and 4 degrees Celsius. This finding suggests that relying on samples stored under less than ideal conditions for EPM diagnosis is acceptable, though refrigeration is still best if environmental temperatures cannot be predicted or controlled during transport and analysis. The study was supported by funding from the Seed Grants for International Activities Program and gifts from anonymous donors to the Equine and Comparative Neurology Research Group. The authors have declared that they have no conflicts of interest and that there was no off-label use of antimicrobials. The study was approved by the University of California Davis-Iacocke, and human ethics approval was not needed. From Vet Neurojar, keep those minds inspired, hearts light, and tails wagging.